Namaste, Hamisha Khush Raho Bitiya. My name is Manisha Singh. Today I am teaching you antimicrobial compounds. Antimicrobial, these are the agents that kill the microorganisms or inhibits their growth. Antimicrobial can be grouped according to the microorganisms, that is antibiotics against bacteria and antifungal against fungi, and the function, that is microbicidals, microbiostatics, etc. Then antibiotics. Antibiotics are the substance produced by the microorganisms which selectively suppress the growth or kill the microorganisms at very low concentrations. Then sources of antibiotics are the fungi, that is the penicillium, Penicillium notatum, that is penicillins, cephalosporins, that is cephalosporium, and bacteria is polymyxin B, tyrothromycin, bacitracin, and cholecystine. Then actinomycetes, that is aminoglycosides, and chloramphenicol, erythromycin, tetracycline. Then quinolones. The introduction is synthetic antimicrobial compounds having quinolone structure. It is bactericidal and broad spectrum antibiotics and primarily active against gram-negative bacteria and most widely used bacteria for their relative safety and their availability in both orally and parenterally and their favorable pharmacokinetics. History is the first member, nalidixic acid, was introduced in mid-1960s, has usefulness limited to UTI and GI tract infections because of the low potency and high frequency of bacterial resistance. In 1980s, by fluorination of the quinolone structure at position number 6 and piperazine substitution at position 7 resulted in derivative of fluoroquinolones. Next is nalidixic acid. It is active against gram-negative bacteria, especially coliforms, E. coli, Proteus, Klebsiella, and Enterobacter, Shigella, and except Pseudomonas. And it acts by inhibiting bacterial DNA gyrase. Pharmacokinetic is orally absorbed, high plasma protein binding, and partly metabolized in liver. And its half time is about the 8 hours. Therapeutic concentration attained in urine and gut lumen are lethal to control common urinary pathogen and diarrhea causing coliforms. Adverse effects are GI disturbance and rashes mostly occurs. Headache, vertigo, visual disturbance are the most common adverse effect. Contradictions in infants and uses are, it is primarily used as a urinary antiseptic. Nitrofurantoin should not be given concurrently, that is antagonism occur. And in the treatment of diarrhea caused by coliforms, norfloxacin and ciprofloxacin is preferred. Then fluoroquinolones, that is, these are the quinolone antimicrobials having one or more fluorine substitutions. Fluorination of quinolone structure at position 6 and piperazine substitution at position 7 resulted in derivative of fluoroquinolones. It has a high potency, broad spectrum antimicrobial activity, slow development of resistance, and better tissue penetration, good tolerability, and used for wide variety of infectious diseases. History is the first generation fluoroquinolones was introduced in 1980s, have one fluoro substitution. In 1990s, compounds with additional fluoro group and other substitution have been developed, further exceeding antimicrobial activity to gram-positive cocci and anaerobes. The classification is first generation and second generation. First generation is norfloxacin, ciprofloxacin, ofloxacin, per floxin. Second generation is levofloxacin, lomfloxacin, moxifloxacin, sparfloxacin, gemifloxacin, and prolifloxacin. Mechanism of action is quinolones target bacterial DNA gyrase and topoisomerase 4. Gram-negative bacteria is DNA gyrase and gram-positive bacteria is topoisomerase 4. And it is double helical DNA and two strands must separate to permit DNA replication and transcription. Overwinding or excessive positive supercoiling of DNA in front of point of separation. It inhibits protein synthesis and DNA gyrase introduces negative supercoils into DNA. DNA gyrase has A and B subunit. A subunit is strand cutting function of DNA gyrase, and a subunit reseals the strand. Then quinolones bind to a subunit with high affinity and interfere with the strand cutting and resealing function, prevents application of bacterial DNA during bacterial growth and reproduction. Finally, bacterial cell death occurs. Hope you all are satisfied with this session. This session is powered by Digital Version 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any query, please mention it in a comment box. I will resolve them whenever we have a live session. Thank you so much for watching this video.